Welcome to the OmicsLogic Metagenomics training program, a comprehensive hands-on journey into one of the most exciting and rapidly evolving fields in modern biology. Metagenomics is transforming the way we understand microbial life, from the human gut to soil ecosystems and disease research. And this program is designed to give you the knowledge, tools, and practical skills to be part of the transformation. I'm on the verge of mysteries, and the veil is getting thinner and thinner. These are the words of Louis Pasteur, the French microbiologist and the chemist who, in the 19th century, pioneered the germ theory of disease. A man so curious, consumed by his curiosity about the invisible world, that even his night felt too short for discovery. That sense of wonder, the feeling of standing at the edge of something profound and unknown, is exactly what drives the field of microbiology to this day. The study of microorganisms, which Pasteur helped establish, gave rise to the tool and approaches that researchers still use and build upon today. Because while we have come across a long way since Pasteur's era, the veil he spoke of is still thinning, and metagenomics is one of the most powerful tools we now have to lift it. For millennia, the smallest thing a human being could see was roughly the width of a hair. Then came the microscope, and suddenly the entire new world revealed itself in our water, in our food, on every surface around us. The observation of a bacterium was made around 1673 by Dutch microscopist Anton van Leeuwenhoek using the microscope of his own design that could magnify the object 30 to 300 times their actual size. This moment marked the beginning of a revolution. The image on the slide captures exactly that. Using helium ion technology and Zeiss Orion nanofab microscope, we can see bacteriophage shown in green actively attacking E. coli bacteria shown in blue. It is a breathtaking glimpse into the war that has been raging on a microscope scale for billions of years, invisible to the naked eye, yet shaping life on the earth in profound ways. Bacteria are not simply isolated organisms, they are remarkably social, able to grow and adhere to almost any surface, forming architecturally complex community known as biofilms. Many of us are familiar with the consequences of these community, even if we don't recognize them as skin conditions like eczema, acne, and dry skins. They are all connected to shift in the microbial balance. These communities are highly interconnected and remarkably resilient. Within a biofilm, the bacteria communicates using a diffusible signal molecule, a process called quorum sensing. Through this chemical, they coordinate defense, adapt to environmental changes, and regulate their behavior collectively. A human microbiota, the collection of all microorganisms living in and on the human body, is estimated to consist of between 10 trillion to 100 trillion microbial cells. Updated research by Sender and colleagues in 2016 confirmed that the microbial cells exist in approximately a 1 to 1 ratio with human cells, with a colon alone harboring around 38 trillion bacteria. But these are not passive passengers. Each microorganism carry important functional properties encoded in their genome that influence human health, metabolism, immunity, and the disease. And this field is still growing, and this program places you right at the frontier of it. So, why does metagenomics matter? The answer lies in the fundamental challenge. The vast majority of microbial life on Earth is simple cannot be grown in laboratory. The traditional microbiology relied on culture growing microbes on plate in controlled condition, but it estimated that 99% of the microbes are unculturable using a standard method. That means for most of the microbial history, we were only ever seeing a tiny fraction of the picture. Metagenomics changes that entirely. By directly sequencing genetic material from environmental samples, soil, water, gut, skin, we can categorize the entire microbial communities without ever needing to culture a single organism. The training program is structured into three progressive levels: the beginner, the intermediate, and the advanced, each building carefully on the last and designed to take you to the foundational concept of all the ways to advance real-world metagenomics workflow. At level 1, which is beginner level, you will develop a solid understanding of microbes and microbial communities and exploring metagenomic sequencing techniques and learn about the sample collection, DNA extraction, and library preparation, and get introduced to the bioinformatic databases and perform hands-on diversity analysis using Data2 pipeline on a real 16S RNA sequencing data. At level 2, intermediate level, you will step into industry standard analysis workflow using KIME2, working with the real Cerasus microbiome dataset. You will then extend into functional metagenomics using PyCrust2 to predict and interpret the functional potential of microbial communities. At level 3, the advanced level, you will master the full shotgun metagenomics workflow from raw whole genome sequencing data through quality assessment, taxonomic profiles, and functional profiling using Human 3. This level is designed to produce research-ready and industry-ready analysts. The beginner level is your entry level point into the world of metagenomics, and no prior coding is required. So you will start building a conceptual understanding of the microbial world and the key principle of the community analysis, and you will explore the different sequencing techniques which are used in metagenomic research and understands how the samples are collected, how the sequencing libraries they are prepared. And you'll also learn about several databases. You'll get introduced to the Data2 algorithm, and you'll learn about the microbial diversity analysis. The Data2 pipeline is one of the most widely adopted workflow in 16S RNA amplicon sequencing, and here you will learn about the sequencing method. The entire pipeline will be considered, which starts from the dataset introduction, the RStudio setup, and the data download. You learn about the error correction, ASV calling, then taxonomic classification, and the alpha and the beta diversity will be followed up here. With your foundation firmly in place, the intermediate level takes you into the tools and workflow that are used in everyday research labs and industry settings around the world. The centerpiece of this level is the KIME2, which is one of the most widely used platform for microbiome analysis. You will work through a complete end-to-end KIME2 pipeline using a real Cerasus microbiome dataset, learning how to execute the workflow, interpret the results, and generate the visualization and apply diversity matrices and analysis, statistical analysis, which are used over here. The second half of this level will introduce you to functional metagenomics, where you will learn about the PyCrust2, which you will be using to predict the functional potential of the microbial communities from your amplicon data. Now, together, these two modules will give you a complete picture of both the compositional as well as the functional dimension of the microbiome research. The advanced level represents the full depth of what modern metagenomics achieve. Here you will move beyond the amplicon-based analysis into the shotgun metagenomics, the sequencing. The entire genetic content of a microbial community will be understood, not just a single marker gene. This approach provides far greater resolution and opens the door to truly comprehensive microbial characterization. You will begin with the dataset download and preparation, and you will learn about the data retrieval from public repository. You'll learn about the quality assessment, then taxonomic profiling, and then you'll dive into the functional profiling using Human 3, which is one of the most powerful available tool for characterizing the metabolomic and functional potential of a metagenomic dataset. By the time you complete this level, you will be able to fully understand, and you will be capable of shotgun metagenomic analysis, and you will be ready to tackle the real research problem. A defining feature of this program is the breadth and the relevance of the tool you will work with. Across the three levels, you will develop hands-on proficiency in the database that power microbial identification through Silva, GreenGene, and others. You will work in R and Python, the two dominant programming language in bioinformatics, and become more comfortable in environments like RStudio and Google Colab. You will use the industry standard quality control and processing tools to handle the raw sequencing data, and you will master the leading pipeline algorithms like Data2, Kime2, PyCrust, Human3. These are not just the academic exercises, these are the exact tools being used in published research and professional bioinformatics workflow today. This program has been designed to be genuinely inclusive, welcoming learners from a wide range of background and career stages. If you're a bioinformatician, this program will help you extend your expertise, especially into the metagenomics tool pipeline and functional analysis workflow. If you're a student or early career researcher in biology, microbiology, bioinformatics, or any life science discipline, this program will give you the practical hands-on computational skills that are increasingly expected in modern research environment. If you're a wet lab scientist who is generating microbial sequencing data and want to take ownership of your analysis, this program is built for exactly that transition. Now, if you are an industrial or environmental scientist, you will find dedicated workflow for your soil, water, and environmental data sets for microbial communities. Now, for the data scientists also and for clinical researchers, these programs engage with disease-relevant data sets, including psoriasis, Rett syndrome, and gut disorders, giving you immediate translation of the skills. Wherever you are coming from, this is a place for you in the program. Let's talk about the outcomes, because the skills you build here are real, measurable, and portfolio ready. Upon completing all three levels, you will have developed end-to-end metagenomics literacy, the ability to understand, design, and execute a full metagenomic analysis from start to finish. You will have achieved a genuine mastery of 16S RNA analysis, the shotgun metagenomic pipeline, functional characterization, the experience of real disease data set, and also you will learn the reproducible bioinformatics workflow. Now, these are not just certificates, these are capabilities that you will learn over here. The OmicsLogic Metagenomics training programs come with a comprehensive learning package designed to support you at every stage of your journey. You will have access to online mentor-guided training program, the asynchronous training program, live Q&A sessions will be there, access to different tools, practice and assessments will be provided, the relevant technologies will be there, and also upon the completion you will receive the certificate which will help you to have a formal recognition of the skills and the knowledge you have developed through this program. So it's not just a training, it's a complete guided experience designed to take you from where you are today and where you want to be in the metagenomics. We are looking forward to seeing you in the program.